Hi everyone, I'm Elena, and today I'm going to be talking about our work looking into sex differences in genetic architecture in UK Biobank. As you know, males and females are different, but what you may not know is just how far these differences expand, which do well beyond the way that we look or our reproductive function. For example, males and females present different responses to particular medications, as well as present different prevalences in diseases like mental illnesses or cardiovascular disease. Despite decades of research and these differences across phenotypes having been well characterized in the literature, we truly don't yet understand the mechanisms underlying complex traits between males and females. A myriad of factors could be contributing to these differences, including our complement of sex chromosomes and our sex-specific hormonal environment. However, differences in epigenetics and genome regulation have also been reported, and in turn, this could also be contributing to some of the phenotypic variation observed. Furthermore, differences in exogenous factors arising from societal gender roles could also be leading to a particular sex being predisposed to a particular socioeconomic status, behaviors, or occupational hazards, and in turn also contribute to some of these differences. However, if we really wanted to simplify it, we could say that there's your genetics and then there's everything else. But everything else is the environment that englobes your genome, and it includes epigenetic differences between the sexes, differences in genome regulation, and all other factors that are determined or influenced by your sex and could thus be interacting with your genetics. As such, given the well-known gene plus environment equals phenotype equation, we could model sex differences under a gene by sex interaction model, where sex-specific factors could be interacting with our genome, thus leading to the differences in phenotype observed. In this context, this project aimed to search for evidence of gene by sex interactions across the genome. Furthermore, in an effort to understand potential mechanisms underlying these interactions, we also looked for links between gene by sex interactions and potential sex biases in gene expression regulation, as well as explored potential underlying biological mechanisms behind the phenomenon. To do this, we employed data from the UK Biobank population cohort, which consisted on around 500,000 individuals of European descent, 530 phenotypes, of which 84 were non-binary, so things like height or hip circumference, and 446 were binary, which essentially consisted on case-controlled disease traits. Furthermore, we evaluated gene by sex across a total of 9 million genetic variants across the genome, which remained after quality control. Whilst this project employed a varied set of methods, at the heart of the project was truly the fitting of sex-stratified models that allowed us to estimate the relationship between our genotype and our phenotype for males and for females separately. Specifically, we fitted sex-stratified linear mix models that allowed us to estimate both general genomic parameters, which estimate the relationship of the genome to the phenotype as a whole, and included things like how heritable a trait is, as well as allowed us to estimate the genetic effects that each individual genetic variant has on the phenotype under study, for each of the 530 phenotypes considered. As such, we obtained sex-specific genotype-phenotype information that we could then compare to assess whether there were differences in the genetic architecture of these phenotypes between males and females. Our results returned a wealth of evidence into the existence of gene by sex interactions. Our comparison of genetic effects across the 9 million genetic variants considered and across our 530 phenotypes found that around 73% of the non-binary traits under study presented at least one SNP with a genetic effect that significantly differed between the sexes, whilst a total of around 10% of the binary traits considered also presented at least one sexually different SNP, which we termed as D-SNPs. Some of the traits that presented the largest number of independent loci across the genome with evidence of gene by sex interactions included waist-hip circumference ratio, height, and diseases like ankylosing spondylitis, gout, or hypothyroidism. We also found evidence of gene by sex interactions in our analysis of more general genomic parameters, finding a good percentage of traits that presented differences in heritability measurements, as well as that presented genetic correlations between the sexes that indicated a certain level of sex-specific genetic architecture for the traits considered. Having found evidence of sex-specific genetic architecture, we wanted to see if we could potentially use the results from our sex-stratified models and our sexually different SNPs to obtain better phenotypic prediction than by using sex-agnostic models, which is what is currently used today to obtain predictions of disease risk. Whilst we found no statistically significant difference in prediction accuracy between sex-agnostic and sex-stratified models, as measured by the correlation between our prediction phenotype value and the true phenotype value in our testing dataset, we did see a consistent upward trend of prediction accuracy across all of the traits considered. As such, this could suggest that as sample sizes get larger and more evidence of gene bisex is uncovered, sex-specific models might be the optimal choice when wanting to obtain accurate phenotypic predictions. Taking this one step further, we look to assess whether sex-agnostic models could be missing loci of interest. For example, a situation could arise in which a given SNP presents a significant association to a phenotype in one sex but not the other, and as such this added extra noise could lead to a non-significant signal when considering both sexes in a sex-agnostic model. We call this phenomenon masking. As such, we looked across the genome for SNPs that were found to be significantly associated to the phenotype in a sex-specific model, but that were deemed to be non-significant in a sex-agnostic model, found evidence of masking across around 22% of the binary and 93% of the non-binary traits considered. These findings suggest the need for studies to perform sex-aware analyses in order to make sure no sex-specific loci of interest are being missed, which could ultimately help explain some of the differences in phenotype between the sexes observed, and even explain why a given sex is more predisposed to certain diseases than the other. Finally, we performed functional analyses in a search for biological insights into the mechanisms leading to the gene by sex interactions observed. These analyses included a gene set enrichment effort by which we transformed our SNP data into gene data, split these into genes presenting a larger effect in males and a larger effect in females. Whilst no overall arching pattern was found, we did find a potential link to mRNA regulation for female-dominant genes in body mass traits. We also assessed whether our SD SNPs were regulating gene expression in a sex-specific way, thus potentially leading to differences in phenotype via the transcriptome. To do this, we used the GTEx cohort's genotype and gene expression data for a total of 39 human tissues. We found a total of 8 SD SNPs that presented evidence of sex-biased regulation of gene expression, but overall larger sample sizes are needed. All in all, we found evidence of sex differences in genetic architecture for a large amount of traits, though be it of generally modest magnitude. We also found a consistent trend of increased predictive accuracy when comparing sex-specific to sex-agnostic models, and found potential masking of loci of interest by sex-agnostic models. Finally, more work is needed to truly understand the biological mechanisms underlying gene by sex. If you want to know more, here is the link to the bioRxiv preprint. Also, our paper has been accepted and will soon be published. So if you have any questions, do let me know, and thank you for listening.